Good afternoon, all of you. And our, hope you all are in a good health and state. And it's literally a pleasure to see you all here, all safe and sound. So I would like all of you to welcome for the webinar being organized by Amity Institute of Biotechnology, Amity University, Haryana. As you can see on the topic, drug discovery in the recent scenario. I would like to invite our speaker for the day, Dr. Chiranjib Chakraborty. Thank you, sir, for being here.
Thank you.
Professor Chiranjib Chakraborty received his PhD degree in 2001 from Vidyasagar University, India. His research work was basically carried out at that time in the Marine Aquarium and Research Center. He did his postdoctorate from the University of Massachusetts, USA. Dr. Chiranjib Chakraborty holds the following position as the Professor, School of Life Sciences and Biotechnology, and also as the Director, Incubation and Innovation Center at Adamas University. He was also a former professor with Galgotia University, Greater Noida, India, and was also associated with VIT University, Vellore, as an assistant professor. Dr. Chakraborty is also a visiting faculty at Institute for Skeletal Aging and Orthopedic Surgery, College of Medicine at Hallym University, South Korea. He was also visiting research fellow at Hong Kong Baptist University, Hong Kong, and also visiting research fellow, research professor at National Sun Yat-sen University, Taiwan. He's associate editor of Frontiers in Pharmacology and a number of editorial boards of scientific reports, Nature, and also interdisciplinary sciences, computational life sciences with Springer. His current research is broadly in the field of structural bioinformatics, diabetes, miRNA, structural and functional biology of medically significant proteins related to disease pathways. Professor Chakraborty has more than 128 Sci publications, 05 books to his credit, 2 edited books, and he has more than 3,000 citations with H index of 30 and item index of 66. So I would like to invite Professor Chakraborty to please share his views on the topic.
Okay. Thank you, Ginny, for giving me the introduction. I'm very delighted to speak in this August gathering. I'm very honored. So, actually, she was my PhD student when I was in Greater Noida. After that, I joined associate professor at the VIT University, Vellore. After that, I Galgotias, and then I have seated here. Okay, I will talk about, I should not waste the time because, am I audible?
Yes, sir.
Yeah, I should not waste the time because time limit is 40 minutes only. So in June, so I would like to start the topic. I think I have shared the screen. You all are seeing that screen, yes?
Yes,
Is it
sir.
okay? Okay. Now, so my topic of the lecture is overview of drug discovery and development using bioinformatics, a recent scenario. Now, in this lecture, you will be able to learn about uh, the aware why and how new drugs are discovered. Secondly, know the process involved in drug discovery in development. Thirdly, you can understand where pharmacologists and bioscientists may involve or may contribute in this process. Fourthly, you can understand the difficulties and danger inherent in the drug development and discovery process. And lastly, I will give about a snapshot view of my work. This is my objective of the lecture. Now, if you can see the question comes in the scenario of what is a drug. Is it a chemical compound like sugar? No. Is it anything which can produce change into the body like eggs? No. So what is a new drug? By defined its characteristics, It can be a potential in the treatment of disease, number one. Number two, it should be a selective in their actions. So these things should be there for the treatment of the disease, and it should be selective in their actions. These two characteristics should be there. Now, the second question is why new drugs are needed. So actually the unmet medical need, for that we need to develop new drugs. New diseases are coming. Previously, we don't know about the new disease like Alzheimer's disease, obesity. We don't know. Now new diseases are coming. So for that, we need new drugs. Some drugs are low efficiency or chemotherapy resistance are there. So we need new drugs for cancer treatment, and several drugs have several side effects, but there is no compound. That's why we or very few compound. That's why we need new drugs. Next is the cost effective of the therapy. Several drugs are high cost, like interleukins, recombinant DNA interleukins produced by recombinant DNA technology. It is interleukin cost is very much high. Erythropoietin, interferon, all these drugs are cost are very much high. Then cost per individual of the country, like TPA, if we can see the TPA scenario, therapeutic plasminogen activator, it is a clot blaster drug. It is also produced by the recombinant DNA process. So if we can purchase that in India, it is more than 3,000 or 4,000 rupees per drug. So per injectable, so more than maybe 5,000 or 6,000 nowadays. So uh, that time I'm talking about like 2005 or something like that, when we was producing the drug. I was in Glenmark or uh, McLeod Pharmaceutical dealing with this type of drug. So that time the drug that cost is like that. So now the pharmaceutical industry's activity, they need the sustainability. For that they need new molecules and patent expiry. Most of the thing is what they have done, the each pharmaceutical company which they have done the patent, their patent is going to be expired near about 10 years. Patent going to be expired, so they knew the new patent. So for that they need new drugs. For these purpose we need new drugs. Now what is the change scenario of the drug discovery and development scenario? If we see the 1800, it was from the natural compound, maybe the plant derived compound. For illness, we can isolate that particular compound from the plant derived compound. That may be the small or organic compound. We isolate that compound, then we synthesize that compound, and we manipulate that compound to get better drug. Like ibuprofen was there, that was that was used for like that way and developed like that way for better drug. So that in that case, there's several problems. That was from the natural resources, limited possibilities prepared by the individual, and that was developed by the small scale and not purified properly and not standardized, no control and no idea of the mechanism at that time. Now we know that all these things has been changed, the scenario and the drug discovery scenario is changed. What is the new modern drug discovery method? You can see what is the difference. The drug discovery method process begins with the disease. So mainly previously it was started with the treatment only. Previously we just started with the treatment. Now we highlight the disease, mainly disease. That from that disease we started the drug discovery process. Now we, after understanding the disease, what is the genetic relevant or what are the biological compound related to that particular drug target, we want to understand, and after that we want to create that particular drug. So the here most important thing is we want to understand or we want to explore the possibilities of the drug target. That is the pinpoint. So here just I have published one article recently. The COVID-19, for COVID-19 we should try for the TLR5 as a drug target, and medical medical virology accepted that article. So we should pinpoint that particular drug target. That is the most important thing for that disease treatment. Now, from that disease, we just understand the biological or genetic target of that disease. After that, we find the lead molecule, or we find the design assay of the molecule, or we found the target of that particular lead molecule of that target, where the lead molecule will bind into the particular target, and will do the high throughput screening after that to understand their activity. Now we want to understand the small molecular head. Then we manipulate the structure to increase the potency. Then we optimize the lead molecule or candidate molecule for a drug candidate. Then we understand the pharmacological properties, the potency, absorption, bioavailability, metabolism, and safety, and then we go for the clinical trial, the 3-phase clinical trial. So this is the drug discovery process for 2000, and you know that it is the synthetic source, unlimited possibility, prepared by a group, and massive scale, so highly purified and standardized, highly legislative control, and mechanism is well understood. So these are the modified method of the drug discovery. Now, if we see the drug discovery's time scale, it cannot happen in a one day drug discovery and development. We should find the time scale. Like, it is a thumb rule time scale. Like, first we have to identify the disease, and from that we have to identify the particular target of the disease. It will take 2 or 3 years of time. Again, we will find the agent of that thing, means we have to isolate the particular protein where drug can bind. Then we will do the preclinical testing. It will take near about 1 to 2 years of time. Then we will go for the IND filing, new drug application, new drug application filing. After that, we go for the human clinical trial. It will take again 2 to 10 years of time. So it near about more than 10 to 15 years of time. If we see that from the identifying to disease to one drug in coming to the market, it will near about 10 to 15 years time. It is a long procedures of time. So getting the approval for India, DCGI, or any country like US FDA or something like that. So if this is the thumb rule for the drug discovery and development process, it's preclinical research 6.5 years. Phase 1 is a phase 1 clinical trial, 20 to 80 volunteers. 1.5 years phase 2 clinical trial is 100 to 300 volunteers. 2 years of time. Phase 3 is like 3.5 years to 1,000 to 3,000 volunteers, human subjects is needed. So it's take near about 15 years of time. This is the general thumb rule for the, and that the phase 1 safety and dose is measured, and phase 2 is effectiveness and side effect is measured, and phase 3 is again confirm the adverse effect is measured. So that's why we get taking the large amount of human subject, that is 1,000 to 3,000 subjects. This is the total drug discovery process. Now, the technology involved in the drug discovery process is identifying the disease, then isolate the protein, the target protein, their genomics, proteomics, and biopharmaceutical things are needed. Something is there. Okay. Now, high throughput screening is there. So we have to screen the compound with the target that is called the high throughput screening, maybe the 1 lakh compound. With the help of the high throughput screening, we can skin with the target and where it will fit. We can understand that. I will show you the docking process, the bioinformatics process, how we can do the skinning of the drug. One snapshot I will show you that. Then you go for the clinical testing of that. Now here, these processes are also involved molecular modeling process. If that protein structure is not available, then you can do the molecular modeling process to get the protein structure, and you can understand where the therapeutic compound will fit with the protein structure. If the structure is available, you can easily discover that thing. Now, identifying the targets, mainly which is the druggable target or druggable genome, that is the big question. Here I am saying the polysaccharide can be a drug target, lipid can be a drug target, nucleic acid can be a drug target, and protein can be a drug target. But problem with the toxicity and specificity, we discard the three categories, that is polysaccharide, lipids, and nucleotide. We discard this. The proteins, we have chosen the proteins are the druggable genome. That's called the druggable genome, and it can, we use the protein as a therapeutic target. Now, if we can see the scenario, one article published by the Drug Discovery Review, that is one scientist has given the snapshot. So GPCR, the most highest group or family for therapeutic protein, the more than 400 proteins are the drug targets are there. The GPCR family is they are more, means all people are concentrating in GPCR family, then kinase, then gene peptide, then serine protease, then nuclear receptor, for P450 enzyme, ion channels, and again other family protein they are concentrating. So they are analyzing mainly the GPCR, kinase, protease, and peptidase family. Now, give me 2 minutes time. I just take water.
So there are a few questions. We will cater them at the end of the talk.
Yeah, definitely. So now the most important thing here, the structural bioinformatics is most important thing, where we understand where the structural bioinformatics can act over there, and where we can how structural bioinformatics can help to understand the druggable target on drug discovery process. The most important question, if the druggable targets have the sequence similarities or domain similarities, or the drug target can bind, where the drug target can bind, and which is the appropriate affinity of the drug target. That is the most important thing. The answer is the molecular modeling. If I told you, if the protein structure is not there into the PDB domain, or PDB mean the therapeutic target structure is not, means therapeutic target structure is not there into PDB, so we can do the molecular modeling, and we can do the docking for the protein computationally. Now, I think the process is all known for the biologists, but who are the science background, some science background people are listening here, they should understand the molecular docking thing. We first understand the protein target, and we do the 3D structure. If the structure is there, structure can be done with the three things, with the crystallography, or or cryo-electron microscopy, or for others we can develop the structure. If the structure is not there, then we can do with the molecular modeling, then we can do the docking with the ligand database, and then we can understand the active site of the protein where the drug is binding with the protein target. Now, like that way, this is the target protein and this is the drug. So here the binding pocket is there. Drug can fit here there and inhibit the particular therapeutic target into the pathway, or activate the therapeutic target into the pathway. That is the docking study. And with the docking study or structural bioinformatics studies, we can do the binding pocket modeling, then lead identification, then chemical library design, the virtual screening, and the lead optimization, binding study. All these things we can do. But the docking study, which is important for docking study, several people are doing nowadays docking study. If you do the docking, any compound will bind any of the drug target. That is the maybe bind with the maybe 10 pocket with the bind. The most important thing, you have to do the energy minimization, where the minimum energy it will find. We have to calculate. We bioinformatician have to calculate that where the compound will bind with the minimum energy, so that the compound will be compound will be bind with the target with most stable thing. That should be there. That is the most important thing. So I can give you one example of the drug discovery using this type of structural bioinformatics purpose, or structural bioinformatics perspective. This is a small story that one company called Aguran, which is a US-based company, who have thought they will means they will search the drug for the HIV. So they found that HIV protease. So then they develop the crystal structure or 3D structure of the protease, and they have developed the inhibitors like protease 1, protease 2 inhibitors, protease 3 inhibitors, several protease inhibitors they have developed, and finally they got one molecule that is called nelfinvir. It is a good overall bioavailability. We know all there things that it is a oral good bioavailability, and it can, this nelfinvir is one of the few drugs for the market which can slow down the HIV or AIDS. You know that probably, and it was developed the structural bioinformatics or the bioinformatics drug development procedure, and it reduce the viral load into the body. So nowadays, for COVID, there are several virus, this type of antiviral molecule. They are trying to use it. The moral story of this, the structural bioinformatics may be the basis for the lead discovery and optimization, but the segment and market size also very important. Segment means the pharmaceutical company are focusing that which are the most important segment size. So if that the segment size is big, they can do the more market, means they will get the more profit. So if you can develop one molecule, it can be easy, pharmaceutical company can be easily taken from you. So, but the fact says that 90% drug entering into the clinical trial are fail to go to the market. They will fail into the phase 1, phase 2, and phase 3 clinical trial. The fifth thumb rule is 90% is failing. The average cost of the drug discovery is near about $770 million. It has been estimated. So these are the general fact. Now the recent scenario. In the recent scenario, I will say some things we are trying for COVID drug development. COVID means the SARS-2. We know the virus is SARS-2. So SARS-CoV-2. So sorry, here one missing a mistake is there. SARS-CoV. It should be SARS-CoV-2. It is not SARS-CoV-2. The drug targets which the people are generally using or finding to drug development are main protease, which is the most important drug target. Nature journal has just give the 3D structure of the main protease, and these protease actually what it is doing, it is keep their means it will keep their non-structural proteins. So if we can block that protein, it will not cleave. So it is a main protease, is one of the good therapeutic target. Then spike protein is, we know that spike protein is binding with the SC2 receptor. The spike protein is also one of the therapeutic target. Another therapeutic target is another therapeutic target is RNA dependent DNA polymerase. So these are the, and you know, so this is, we have developed some, we have tried some making some molecule. The time limit is short, so that's why I am here. I am just here. Some some uh, vaccine related, in silico vaccine related, we have also done and done for, we published in medical virology for vaccine related thing, and we have also done the aeromolas vaccine in silico. We have also done the you know, uh, you know in silico vaccine for the prostate cancer. We have done the molecular dynamics and muscle proteins also mutation. Okay, thank you. The time is short, so I cannot
So
take
we
more
can
time. I
still
should
take
not take.
some questions.
Okay, we can take. I should acknowledge my group. Yes, we have a big group. So Murthy, Lee and Win, Zoo are there. Then from Asis, Garima from India, they are in Halim University, South Korea. And then Manojit is there. He's also in Fakir Mohalim. So my take home message is closely watch to the drug target and listen to the cells about their problems to silence the disease. Thank you for kind attention.
Thank you so much, sir.
Okay.
So the questions are, the first question which was asked by Mr. Manoj was, can you please elaborate about filing IND?
So actually, India has the DC control of DCGI, controlling the DCGI. So DCGI, I think for uh, clinical trial, Form 44 is there. So for each clinical trial, there is a procedure you have to submit for each clinical trial. Then finally, after phase 3 clinical trial, you will get the approval. The each form is there. You please go to the DCGI site. You will find their form. You have to fill up that form, and there is a fees like 50,000 or something like that. So with this, you have to apply drug, like it may be a stem cell, it may be a recombinant, or anything you have to apply. They have given the definition of the which is the new drug. If it is a drug, so you have to do the 3 phase of the clinical trial. For that, go for the 3 phase, and all
So your voice is not
them,
audible.
and they, okay, is it audible?
Yes, and now it is audible.
Okay, so with the help of the form 44, you have to apply that, and with that form 44, you can apply to the DCGI, and every country has each own regulatory body. So they have the, if you want to market that drug in like USA, you have to apply the US FDA, or Australia, TGA Australia. So different country has a different regulatory body. So you have to apply that. They go their side. I should request you go their side. They have a definite form, like for India, form 44. So they have a definite form. So you have to
Okay, so
apply
another
that. Okay.
question is how
Yes.
to find the lead compound hit. And similar question is how to find the lead compound hit in molecular docking. And to basically elaborate about
So
scoring.
scoring system, is there, but you have to do you have to dock random docking. You for docking, you have to uh, do with good software, not the free software. You have to like 100 draw or 100 dock or like that way. You have to try with a single molecule, and where you will find the minimum energy docking, that place you have to choose, and then you have to understand which are the bonds or hydrogen bonds are there. uh, all these things you have to choose, and then it will be the best docking thing, and then only the paper will accept your view. The high impact factor journal will accept your view, because you have on the energy side you have to explain with the energy side, with the minimum energy thing, so that the docking will be the stable thing.
So
Next
another
question.
question, I'm so sorry, sir. There are numerous questions, actually, number of questions which are lined up. So if you can be a little quick in answering those. How
Yeah,
drugs
yeah,
are different from
yeah.
vaccines?
No,
That's one of the
vaccines.
question
No,
by.
DCGI has given the definition which are the new drug. So if it is a new thing means if it is a new compound which is not approved any country, so it is a new drug. The DCGI has already given their side. It is a new drug. So if you, on the other hand, if you can like any compound which is approved for a particular symptom, and you are a symptom, you are trying to apply in another symptom, like the compounds which are applying antiviral drugs which are approved for the AIDS which are applying into COVID-19, it will be treated as a new drug, and you have to go for the clinical trial. Like that way, DCGI has given all these definitions which are the new drug. Like that way, the biological compounds which are new, like vaccines, recombinant DNA technologies, pre-protein, or any stem cell, they will be the new drug. It is not a different from a uh, general compound. It is also it will be for DCGI or pharmaceutical point of view, it will be new drug, and you have to go same way which the drugs and molecular act is there. You have to do the 3 phase of clinical trial, and if it is known, means known compound, and then only for other country, if you going there, so you have to do the 3rd phase of clinical trial. So
Sir,
this is
another
next
question is,
question.
yes, sir. Sir, by Mr. Joydeep, he's thanking you for the nice presentation and saying that since we are running out of time in the fight of COVID-19, what do you think will be more effective strategy, either to go for repurposing of older drugs rather than development of new drugs, and what will be more effective strategy?
No, definitely repurposing of the general drug one we have to, but again there is a DCGI rule. We have to DCGI, and every country's rule, we have to go for the 3 phase of clinical trial. But the drug controller general or if of each country can shorten the clinical trial with a minimum amount of patient, and repurposing of the new drug with the shortest route. And the vaccine development also, vaccine development can be done from the bioinformatics, and after that, it can be developed through the wet lab procedure quickly.
Yes, sir. Sir, another
Okay.
question is, uh, how RNA-dependent RNA polymerase can be a target source of SARS-CoV-2?
Uh, one drug is already approved by the remedies with, you know, that it is with the means it is targeting the RNA dependent RNA polymerase. When it will target means for the COVID we don't know the particular mechanism, but for in general the virus, like it was approved the Ebola virus, so we know that virus, it just inhibit the viral synthesis. So when it will inhibit the viral synthesis, the viral load will be less into the body. So that
So,
way it is working.
yes. So one question is about energy. So one question is about energy minimization. So the question is, if protein binds with any molecule and we do the energy minimization, what is the cutoff? When can we say that binding is good enough?
So the minimum energy we have to think about that, the minimum energy. I don't know. We are trying with the minimum energy. Cutoff value we don't know. Like docking procedure, we are doing the docking procedure, maybe 100 docking. After that, we will find the minimum energy. We generally scientists way. Cutoff value we don't know. For each molecule, it may be different.
Sir, we have again a number of questions which are still left over. So I'm just trying to pick them up as fast as possible. So Mr. Manoj Kashyap has said that Professor Chiranjib, fantastic talk, and I would like to know what is the success rate for drugs identified using docking and bioinformatic approach, and they actually went into clinic eventually.
That rate I don't know, but with the virtual screening you can search with the thousands of molecules at a time, dock with that, then you get the better compound, and it will means actually with the help of the bioinformatics the time will minimize. What you will have to find out with the laboratory, if we can do with the laboratory, do the testing, it will take more time. But if we can do the with the bioinformatics, it will the time will be less, and then you ultimately you have to do with the mice study and animal modeling study. After that, you go for the clinical trial.
Okay. So can nanomaterials be used for targeted drug delivery at specific disease site? This is a question being raised by Mr. Abhishek Chatterjee.
Yes, definitely. Targeted delivery. That's why the nanomaterial are using. But again, it will be if like rabiprazole. Rabiprazole molecule is already there. If we can do the with the nanomaterial so release molecule, like it will act whole day, so it will be treated as a new drug. When it will be a new drug, you have to go for the 3 phase of clinical trial, all 3 phase of clinical trial. So that is the thing is there. So nanomaterial you can definitely use, but the thing, if you change any molecule with the help of the nanomaterial, the effective drug delivery, as per the all regulatory body, like all countries regulatory body, it will be treated as a new drug. So you have to go for the 3 phase of clinical trial again.
So another question, another actually statement given by Momita Mukherjee. She's saying that Dr. Chakraborty, very informative discussion. And there are scientists from SN Bose Center which are proposing for nanomedicine. Any prediction about its success?
No, I don't know about the nanomedicine. I have edited a couple of papers as the editor in Frontiers in Pharmacology, but I have not done anything about the nano. So I have not detailed knowledge with the nano drug. So I cannot give you a
Okay, sir.
particular
Sir,
hint or success rate.
one last question for the session. Generally, researchers use free software Autodoc. Please elaborate whether Autodoc 4 or Autodoc Vina is good for virtual drug screening. This is a question by Dr. Manish Chandelier.
Yes, you can use it Autodoc, but if you can, you know, take some good software, like different pay softwares are there. So with that help of the softwares, you can means you can analyze several other like energy minimization, several other factors also you can analyze. Uh, so that your docking can be the successful. Autodoc you can use definitely, but the
So
thing
he's
is
asking Autodoc 4 or Autodoc Vina.
Autodoc Vina maybe is
Oh.
one of the good choice.
Thank you so much for this elaborate discussion, and you have taken up a lot of questions which include our faculty members and students. Thank you so much, sir, again for being a part of
Any if you have
this
any
webinar.
specific question, you can write to Dr. Jini Tomar. He will send me. I will try to I will able to give you answer of the question in the future. Definitely, if you need any help, you can ask me in future also. I will help you if I
Sure,
can
sir.
do that
I'll
also. Definitely within my limit.
share your email ID with the group. And thank you again so much. Thank you to Amity University for enabling the organization of this webinar. Thank you, Chiranjib sir, to all the participants who have
Thank you.
patiently listened to our webinar. Thanks again. Be in touch, stay safe, and please keep smiling. That
Thank
is
you.
something which
Thank
is
you
very
all.
required. Okay, thank you so much.
Okay, thank you. Thank you all of the, all of you to listen to my talk. It is my pleasure and I'm grateful to all of you. Okay, thank you.
Thank you, sir.
Thank you,
Okay.
sir.